Hey guys, how you doing? This is just a mini crash course on electrophoresis and general parts of how it relates to DNA and whatnot. Um, Quick little side note: this was these two guys, O.J. Simpson and President Clinton, were two of the first examples of DNA science making its way to the general public in the United States. The first was the murder trial of O.J. Simpson. I think it was '95, and the second was uh, President Clinton, and I think it was '91, '92, his alleged affair with an intern, and there was some material found in the scene on a blue dress, and whether that material belonged to the president. So these are the first two major samples or examples of when this science was kind of first hit the American public. So let's take a look at how we can do a DNA fingerprint and how it relates to electrophoresis. So let's take, for example, Jay-Z and Beyonce, and let's try to see if we can figure out how we might determine whether two children were theirs or not. So the first thing we have to do is we have to go into the same chromosome of both of these individuals, and we have to find a particular area, and you'll see here these are actually marks which would be equal to restriction enzymes, which are enzymes that cut DNA. And we see here that at this particular location, Jay-Z has GATC repeated 4 times. So we call them short tandem repeats. And it's a way for us to tell individuals from each other. And so we see here that this restriction enzyme is the same, GGG, TG, TCT, and the same down here. So it's going to cut the same front part of the DNA, and you can also see that we use the same restriction enzyme on the other side, which gives us these differences. So we do know that every human, of course, has 2 copies of each chromosome. And so being a diploid organism, we're just going to say that Jay-Z happens to have an STR of 4 repeats on both chromosomes. So we consider him what's called a 4-4. Beyonce, on the other hand, we're going to make her a 5-6. And basically that means that she has a repeat of GATC 5 times on one chromosome and 6 on the other. So if they were to have a kid together, and this is not their kid, but I just wanted to find one that might look something like their kid, you should be able to figure out what gene possibilities, what STRs this kid should have based on if Jay-Z and Beyonce are his parents. And so we're just randomly going to give him obviously a 4 from Jay-Z, because no matter what, whether it's this chromosome or the other one that goes in the sperm of Jay-Z to make him, it's going to be a 4, and we're going to call him getting the 5. And let's just say they had a daughter. Again, this is not their kid, but if they did, we would see that she gets the 4 and the 6. So now, how do we know that? We basically know that by looking at the DNA and taking the DNA samples. And so electrophoresis is one of the ways we can do it. So what is it? The big nerdy book answer is a separation technique using electricity to run molecules through a porous matrix to separate them. This is the idea behind it, that the speed is inversely proportional to the size. So the larger the molecule, the slower it goes. And let's grab an example of that. So let's talk about this as being electrophoresis, gel electrophoresis. And we have 3 fragments of DNA that we're going to put through it. So here's fragment 1. Is this fragment going to have any trouble going through this gel matrix? And the answer is no. This little dude's going to be able to zip through, no problem. Even this part that's tough, get up there, not a problem at all. What about this person? Now, she may be stronger, but obviously she's larger, and so she's probably going to have a little bit of degree of difficulty getting into some of these tighter corners. What about some of my favorite rappers from the 90s, Big Pun and Fat Joe? Clearly you can see here that these guys are going to have a very difficult time moving through this gel matrix, and so therefore we can use this as an example to understand that DNA fragments move based on their size, and smaller fragments move faster than larger fragments. So this is what the agarist looks like, and we've used this in lab, and these are the wells that we use to put the actual DNA in. And if you look under an electron micrograph, this is what the gel actually looks like. So you can see all the holes, and this actually allows the molecules of DNA to move through. So once we have used this technology, we can see here that in a certain lane, the lane was poured here, and we can see that the DNA moved. Now, even though each of these fragments looks exactly the same size to the human eye, they're not. This is not one fragment of DNA. This is billions of fragments of DNA that all have the same size. And so if you look at it that way, this little fragment here is definitely the smallest piece of DNA, and this fragment here is the largest, and so it went a lot slower. And so we can use that technique to separate these molecules. So let's go back to this sample. Here we have Jay-Z and Beyonce, and here's our two kids, and we don't know what they are, and let's see whether or not they're actually their kids. So the first thing we do is take the DNA, extract the DNA, and we would use the same restriction enzymes in the same locations with the parents as we would the kids. And so when we run the gel electrophoresis, we get this. So if you recall that in electrophoresis, the larger fragments move slower. Let's see if you can figure out what this band is and what this band is for the girl. So you should have got an answer of, in this case, 4, and in this case 6, because if you remember, the girl was a 4-6. The boy was a 4-5, and you can clearly see that both of these are on the same plane, right? And also we see that this 5 is halfway between the 4 and the 6. So these fragments can, obviously we have to be able to attribute them to both of the parents. And of course in this case, we can answer the question of, would this third kid, could you attribute the possibility of this third child as being an offspring potentially of Jay-Z or Beyonce? And the answer is there's no way this kid could be the offspring of Jay-Z, because Jay-Z only has STRs of 4 at this location, and notice that this child does not. But the kid could be Beyonce's kid, because you can see here that the 6, this kid also has a 6 and a 7, and looks like a 10. But the kid does have a 6, so it's possible that this kid is Beyonce's child, and I think Jay-Z would be a little pissed to hear that. So this is an example of what it looks like for real. This was some of the earliest work on DNA fingerprints. It was done with the Romanovs. If you guys don't know the Bolshevik Revolution, we can see here that the marks here, the blue bands, are actually DNA sequences. They're STRs of the bodies that were found at the gravesite. And we can use this example, just like with Jay-Z and Beyonce, to see whether any of these kids found in the gravesite are actually the Tsar or the Tsarina. So we see here that the Tsar has a band here, and here the Tsarina only has 1 at this particular location. So that means that all 3 of these kids better get every band from mom and dad. If there's any extra bands, then one of their parents is not their parents. So if we just do this, for example, with child 1, we see that child 1 has a band here and the Tsar has a band here. So the child 1 has his second band. Well, where did that come from? If we look over here, there we go. So that's one of the ways that we know that the Tsar and the Tsarina are the parents of that kid. The last little part I want to talk about in this video is the amounts of STR locations. I might, for example, if you were to take an STR of me in the same location, I might have a band here and here, but it doesn't mean I'm the offspring of the Tsar and the Tsarina. So when we do this for DNA fingerprinting, we use several different locations, and then we add up the mathematical chance of how many people would have those locations all identical, and that's how we get to the science behind DNA fingerprinting. Hope it helps.